So welcome everyone to the September Soils Network of Knowledge webinar. If you're listening but you're not a subscriber, here's the link to subscribe to the webinar. Now I'd really like to introduce Brian Murphy, who's presenting today's webinar, and the title is Understanding Soils and Interpreting Soil Tests. What do all the numbers mean? Bit of background on Brian. He's worked as a soil scientist for more than 30 years, going back to the Soil Conservation Service on the agricultural, conservation and environmental aspects of soil science. He is a co-author of a book called Interpreting Soil Test Results, What Do All the Numbers Mean? His experience has included the production of several soil maps for major parts of New South Wales, and he's been involved in research on the effects of land management practices on soil carbon and soil properties. Brian's also been author and editor of 2 published texts on soil science and was a major contributor to development of the Land and Soil Capability System in New South Wales. Throughout his career, Brian's worked on the on-ground application of soil science to develop and implement better land management practices. And Brian's still attached to OEH, Office of Environment and Heritage, and ANU.
Okay, the first thing I was going to talk about today is the New South Wales Soil Knowledge Network. And it sounds very much like the other network, but the New South Wales Soil Knowledge Network is made up of a group of 12 retired soil specialists from around New South Wales, and essentially they're used to all work for soil conservation or the organisations such as Office of Environment and Heritage and Land and Water, which are all derived after soil conservation. And one of the reasons we've got together as a group is that in recent years there's been a bit of a loss of soil specialists through retirement and redundancies. So through a citizen science project run by Sally McGuinness-Clark, this Soil Knowledge Network has been set up. And there's just a few, it's not for profit, so I don't go out trying to make money or anything, but it's mainly a resource to try and provide some advice and act potentially as mentors and trainers to other soils people who'd like to know more about soils, and not only soils people but all land managers. Yeah. Some of our projects that we've been involved in, one is trying to get an effective state soils policy. The other one is we've done an educational video series, and if you look on Facebook, and I think even if you just go to Google and log in Soil Knowledge Network, you should be able to find those videos. And the videos are on things like the difference between sadic soils and saline soils, and how to look at soils in the paddock and a few things like that. We've also been involved recently in some technical workshops with agency and community partners. And the other job we've been working on is the release of development of some soil landscapes map for the Central West. Just a couple of photos. The photo on the top left was where we had a project, I think Roy Laurie and Sally McGuinness-Clark did some work with school children, and here's just some of the other field trips which we've been on looking at different soils. Right. So now I've told you about the Soil Knowledge Network, and certainly if you want to be involved with the Soil Knowledge Network, you know, just contact Sally McGuinness-Clark or myself. Yep. So the topic of today's talk is about understanding soils and interpreting soil tests. The reason for interpreting soil tests, one is for soil mapping, the other one is for regional planning and investment plans and catchment management plans. We look at land capability and land degradation risk, but you also look at land suitability and the productivity and suitability for specific purposes. The other thing we use soil tests, of course, is for specific paddock site information on acidity, nutrient recommendations, and problems like salinity, sodicity, soil structural problems and contamination. And at the end I might just talk very briefly about soil sampling. Now, interpreting soil tests, I think it's important to remember what your resources are, and one of the best resources around is a book by Peveril, which Peveril comes from CSIRO, I think, and it provides for all the different soil tests and for different soil properties. This book provides the background and theory behind a lot of the sampling and measurement and interpretation, and it has interpretation tables for a wide range of soil properties, including soil physical properties and pH, aluminium, electrical conductivity, cations, sodicity, all the nutrients, and the trace elements. The issue with this book may be that some people might find it a bit heavy because it does go into a lot of background and so on. The other book, which is useful for interpreting soils, is one by Hazelton and myself, and it also has a wide range of interpretation tables for different soil physical and chemical properties, but it also includes some engineering properties and a bit more emphasis on environmental properties in relation to erosion. So, but it's nowhere near as theoretical and doesn't go into as much detail as does the Peveril book. So in terms of soil mapping, we do soil tests, and if you look up here you can see this landscape here is called Merry, and we know that soil landscape has lots of surface sodic soils, which is a problem. We know that these dark soils here in Caribbean, which is the yellow bit here, we know these dark soils, which is the cracking clays, they also have some problems with surface sodic soils, but not as bad as Merry. And the soil map, we're also, by doing the soil test, we know that this area down here called Lower Bugwa has lots of salinity problems, but it also has lots of scalding. And if you know anything about the scalding north of Nyngan, this is where all that scalding is. So the soil tests are very, very important for helping us interpret soil maps. The other thing is land capability. And the things we look at in land capability are erosion problems, acidity, structural problems, salinity, and we also look at things like rock outcrop and waterlogging. So all the soil tests is helping us to understand about land capability. Class 2 land here you can see is very good for cropping, but I know this particular soil landscape and we know that it has some soil acidification problems. Class 4 land is good for grazing, and you can see Class 7 land is good for trees. Yeah, all right. And just for interest, you can see this area here of very good conservation farming or conservation tillage practices, double retention and minimum disturbance at sowing. So that's very good for erosion control and building up soil organic matter levels. And you'll notice here on this sloping land which is being tilled, under a rainstorm there's obviously erosion. So one of the first things about soils is you've got to know the general soil characterisation, and the important things are texture, the colour, the structure, and you should look if you know there's self-mulching soils, that's very important because you know that those soils are going to be good. If there's very strong lenticular structure in the subsoil, you know you've got lots of shrink swell problems. The other thing which is important is soil organic matter. The other thing that's important is the response to wetting, whether it slakes and disperses, so whether the soil structure breaks down under wetting. And the other important thing is the depth of the soil, the depth to the rock, the depth to saline layers, or maybe the depth to dense sodic subsoils. And this is a texture triangle, and texture is a very useful and underrated aspect of soils and important. So you should learn how to do field texture because that will tell you a lot about how to manage your soils. Sandy soils have to be managed quite differently to loamy soils and to clay soils. In the lighter textured soils, soil organic matter is really important for both cation exchange capacity and for soil structure. In the clay soils, it's the exchangeable cations which control a lot about the soil structure, and also the type of clay determines how much cation exchange capacity you have. So if we have a look at some individual profiles, this one here is a red chromosoil. It has a fine sandy loam topsoil over the red clay B horizon, and you can see the lucerne rip there quite likes the B horizon. Whereas this soil here is a soda sol, and you can see the bleached horizon, which is caused by the very dense yellow sodic B horizon. And I haven't got the ESP's exchangeable sodium percentages, but I think they were around about 18% from memory. So if we're going to interpret soil properties in the soil physical properties, the things to look at are water holding capacity and the plant available water, the aeration and bulk density, which is very dependent on texture, the soil strength, the potential for surface sealing, which affects your germination and emergence, and the infiltration and runoff. So texture effects, now this is plant available water, which is the difference between the field capacity and the permanent wilting point. And of course clays hold a lot more water, but there's not as much water available to the plant because it holds the water so tightly. You might, sandy loams have about 16 millimetres, fine sandy loams about 20, but the two which have lots and lots are the loam clay loams and silty clay loams. What's also interesting about that is that lots of the Russian Chernozems and the black soils on the plains of the US where they grow such terrific crops tend to be in this group here with this very high water holding capacity. The limiting bulk density is different for different textures. Sandy loam, the limit is 1.8. For clay, it's 1.4. And the reason is because of the amount of air in the soil. Sandy soils are much more open and can have a lot more pores, so they can have a higher bulk density without limiting plant growth. Clays have fewer large pores, so their bulk density limits it much lower. Just to give you an idea of what happens as soils dry, at the dry end of the scale, soils are too strong, and 2 megapascals limits root growth. At the wet end, if aeration has to be more than 10%. So as soils go from dry to wet, you get a nice non-limiting water range in here if you've got a good soil structure. However, if you have a poor soil structure, which happens with sodic soils and sometimes under compaction, you actually get no non-limiting water range. So roots find it very difficult to grow in soils like this, because that's, by the time they lose enough water to increase their soils to have air available, then they are too strong to allow root growth. At the same time, when the strength of the soil becomes less enough to allow roots to grow, there's no air. So that's a problem. For soil chemistry, the things that are important are cation exchange capacity, pH, and I should mention here that pH less than 5.5 in water causes aluminium toxicity and may affect P availability as well. But if you have a pH of 9, more than 9, you've probably got hydrogen carbonate around, and that will only happen if your soil is dominated by sodium and you've got a sodic soil. Then you've got electrical conductivity, soil organic matter, nutrients, and the toxicities. So I'll just go through those. Exchangeable cations. Clays have a residual negative charge because of their structure. It's just the way they're built and the way their atoms are made. Therefore, you've got to have exchangeable cations to balance that residual charge. And they're made up of sodium, calcium, magnesium, and potassium, and sometimes aluminium. Sodium, if it's more than 5 to 6% of the exchangeable cations, causes dispersion. Calcium is very good for the soil. Soils high in exchangeable calcium provide stability to soils. Magnesium is less effective in providing stability. Has only about 60% the flocculating effect of calcium. So sodium magnesium soils are have much lower structural stability than sodium calcium soils. Potassium actually causes dispersion, but it's not very common. Aluminium only really occurs when the pH in water is less than 5.5, and when the aluminium ions are in solution, they become toxic to root growth, and that's one of the big problems with acid soils. So cation exchange capacities, units are centimoles of positive charge per kilogram. You can see 6 is low, 6 to 12 is, sorry, less than 6 is very low, 6 to 12 is low, 12 to 25 is moderate, 25 to 40 is high, and greater than 40 is very high. Because these would be mainly sandy soils, and they have a serious problem because it doesn't give them much buffering capacity against acidification, and they can't hold much nutrients. These soils here greater than 40 are normally your smectite clays and the black cracking clays of the Liverpool plains. just to give you an idea of how much cation exchange capacity there is in each of the different types. Coarse sand virtually has none, fine sand none, silt maybe a very tiny little bit, kaolinite clays have a little bit, illite clays, which are really common in a lot of the red chromosols, and it's certainly in a lot of the, even the cracking clays in the Macquarie catchment. And the spectite clays are the ones which have really high cation exchange capacity. And the spectite clays are very common from basalts. The other important component of cation exchange capacity is the organic matter, and it can have 150 to 500 centimoles of positive charge per kilogram, depending on the organic matter. The humus has the highest value, so if you've got good humus in your soil, that can add a lot to your cation exchange. As I mentioned, in this lighter textured soils like sands and fine sandy loams, organic matter can be a very important part of the cation exchange. And just to give you a visual, these are the clay plates. Here, a clay plate here. There's the cations here, sodium, calcium, magnesium. And what happens when there's water around, sodium likes water, and because it's only got one charge, there's a lot of sodium atoms, and they all like water, so the water rushes in and pushes the clay plates apart, and you get dispersion. The controlling factor on that is that these are the exchangeable cations, which are intimately linked with the clay plates. Outside, you can have salt, and when you measure the electrical conductivity, you measure what is effectively, the free salt outside the clay system. If there's a lot of salt outside here, obviously that's going to reduce the free energy of the water, so the more salt you have outside here, the less water that will rush in to push the clay plates apart. So one of the ways of getting stability is to add salt. I'll talk a bit more about that in a minute, a bit more about that. Here's some of the problems. You can see surface sealing, problems with emergence and germination on this sodic soil. Huge cloddy. You can see how cloddy that particular paddock is. Here's another problem. If you get subsoils, can be extremely sodic, and this one I think had the ESP of around about 32. That's a person just there. So you can see there's severe gully erosion. That's in the Bathurst granites. The other problem you get with sodic soils and dispersible soils is you can see there's a dam, right? But it was a dam, but now it's got a great big hole through the middle. So that's caused by dispersion in sodic soils. I might just skip that and move on for the course of time. Okay, managing soils and acid soils. So if the pH in calcium chloride is less than 4.7 or less than 5.5, there's aluminium 3+ in solution, which is toxic to plant roots, especially to sensitive plants such as lucerne, some wheats, chickpeas, phalaris, and there's probably quite a number of other plants which don't like aluminium in solution. And the question is, why pH in calcium chloride and not in water? It's because pH in calcium chloride is the most reliable and consistent measurement. pH in water is affected by the soil electrolyte concentration and can vary seasonally by as much as 1 unit, 0.5 to 1 unit. Whereas by adding the calcium chloride, the pH values are stabilized, and that is very much what the plants see as well because of that. So usually when you're trying to manage acid soils, you'd use a pH in calcium chloride rather than pH in water. And adding lime is the way that acid soils are often managed. Here's a big pile of lime. Here's a spreader adding lime to the soil. Electrical conductivity. There's a saturated ECE, and then there's a 1 to 5. We can convert one to the other because, as you see, this is one part of soil to 5 parts of water. This one is saturated ECE, so that means the soil is saturated. So how much water the soil holds at saturation determines what the conversion factor is. So if you don't have much water, as the case with loamy sands, then you have a much higher conversion factor. If you have quite a bit of water, as you do in clays, then you have a lower conversion factor. The units are in decisiemens per metre, microsiemens per centimetre, and total dissolved solids. And of course they all appear in different publications and cause a bit of confusion because people quote numbers and often don't mention the units. Effectively, I think microsiemens is about 1,000 times decisiemens per metre, and total dissolved solids depends on what the salts are because they all have different conductivities. Like sodium chloride, I think, has the highest conductivity, whereas carbonates tend to have the lowest. And the salinity, if you see here, salinity greater than 1.5 decisiemens, and that big saline patch there, those soils would have an EC greater than 1.5 in a 1:5 extract and greater than 4 in the saturation extract. And nitrogen, right? And nitrogen, okay, nitrogen, when you measure nitrogen, you measure ammonium, you measure total nitrogen as soils, or you measure ammonium and nitrate. The issue about nitrogen is that you can measure total nitrogen, which is nearly all organic, or you can measure the nitrate. And just some numbers, total nitrogen, if it's less than 0.05, then that's low. High is greater than 0.05. Nitrate, there's some numbers. The other aspect about nitrate is that toxic as it gets into drinking water. I just might briefly mention phosphorus, because phosphorus can be in crystalline surface absorption or in organic matter, and when you measure phosphate, you try and use an extraction using bicarbonate or lactate or fluoride, and the phosphorus extract actually takes this part out, and some of the surface adsorbed phosphate. So and typical values for soil phosphorus depend on the buffering capacity of the soil. And I should go back to the other slide. The amount of phosphate that's held on the surfaces, crystalline surfaces, especially iron and aluminium, determines your buffering capacity. So if you have a basalt soil, you have a high buffering capacity. If you have a non-basalt soil, you tend to have low buffering capacity. And they're typical values for what your phosphorus level should be. You might notice if you've got vegetables, you need more phosphorus than if you're trying to grow a dryland pasture. And soil organic matter, I think the critical thing here is that generally from numbers from England suggest if you've got more than 2% soil organic carbon as grams per 100 grams, your soil's running nicely. If it's got less than 2%, then it's probably not functioning as well as it could. And interestingly, if you look at some of the numbers which come out of Australia, there's a lot of Australian soils which have less than 2% soil organic carbon, especially under crops and under pastures. The other thing is that you've got to make sure when you distinguish between soil organic carbon and soil organic matter. Now I guess this last point here is about soil contamination. And soil contamination, the actual critical levels depend on what you're going to use the land for. If you're going to use the land for, say, playgrounds and playing fields, then you have a different value. And this one here is for home, where you might have preschools and primary schools and children picking up soil and putting it in their mouth. Your critical values are much lower than they will be for industrial. So that's why setting critical values up is not simply as straightforward, oh, that's one set of values, we'll just keep those. It depends on the use you're going to put the soil to. I just might mention a thing about sampling soils. When you sample a soil, you actually sample a great big volume of soil like this and then get a mean value, because the root grows in that little bit of soil there and only sees the soil which is sort of adjacent to it. So sometimes soil tests don't line up that well with what you see. The next thing is about sampling a paddock. And I think some people say you should take a transect across the paddock and then use that mean number. But you can see you miss a lot of the paddock, and there's probably a good case for saying you should use a zigzag pattern to sample your soil. Even though you zigzag pattern, you might take those samples and get the bulk sample from those. It's still better to take a zigzag pattern than a single transect. So just conclusions. Purpose for sampling and reason for obtained data needs to be well understood and clearly defined. Soils need to be adequately and effectively sampled for the purpose. You need spatial coverage, and some purposes you might need a randomness so you can do some statistical analysis. The laboratory measurements need to reflect and imitate the processes in the soil to provide information on the performance of the soil. Interpretation of data and tables of critical values are usually derived from planned experiments and trials or from a long history of experience. But obviously if you're outside the experience that people have used or outside the experience of the trials, sometimes the data mightn't be that accurate. I guess as a final conclusion, sets of interpretation tables of critical values for different soil properties can probably be thought of as the language or alphabet of soil management. They can be used to put together narratives about the capabilities, productivity and limitations of soils. So it's just an interesting way to look at soils. However, Albert Einstein said something that said, not everything that counts can be counted, and not everything that can be counted counts. If you go into a paddock and this is what you see, I don't think you need a soil test to tell you that that soil is probably sodic and you've got big problems. This soil here is actually a Russian Chernozem. You can look at that soil and say, I would say that soil has probably got a lot of good things going for it. And so you probably don't need a soil test to tell you that. If you look in a landscape like this, you can say there's lots of good things. That's a good soil and it's highly productive, and it might have some problems, but it's basically a good soil. So I think that's where I'll leave it. Yeah.
Fantastic, Brian. That's a big ask, wasn't it, to try to cover all of soil testing in 20 to 25 minutes. So great effort. Julie Dart, I can see, has her hand raised.
Okay, Brian, the question I've got is, up this way we tend to have labs that put out alternative testings like Reams and Albrecht. Just wondering what we can do with those results when we get them, because often they're standardised against different things and it's hard to know what to do with them.
Yeah. Yeah. Well, all the tests I talked about were done using the standard procedures, you know, as in the Rayment, well, it used to be the Rayment and Higgins book, but there's a new one out now as well. I think that's always a difficult question to answer, because they often use different tests and use different extraction methods and so on. And I think one of the things I mentioned was, if you're going to interpret soil tests and use soil tests, then the way you do the soil test should reflect something about how the plant sees the soil, or at least reflect how you're going to use the soil test. Like if you're going to use the soil test to predict how an earthwork performs, then you try and relate that soil test to the processes which might cause the earthwork to fail. So you should, if you're using it for calcium-magnesium ratios and so on, you should think about what's the soil processes which you are trying to talk or make interpretations about. And the other problem you have is that sometimes interpretations can come from, say, sets of soils which are very different. And I think I made that point, that people will do a set of trials and set up some experience and have some, you know, interpretation values, but they base that on that set of trials and their experience. If they move into a different set of soils, then their numbers might not be so useful and so easy to interpret. And one of the things I probably should have mentioned was that the most valuable person in trying to interpret soil tests sometimes can be your local soil scientist or your local agronomist, say from Incitec or someone, because they probably have the local knowledge to say, okay, I know this soil test means this on these soils because they've had the experience. So I'm not sure if that answers your question about those alternative methods. I don't honestly know about a lot of their methods, But I think, I know they use calcium-magnesium ratio a little bit, but I think there is the calcium-magnesium ratio is very important from the point of view of soil dispersion because, as I mentioned, magnesium only has about 60% the flocculating power of calcium, so sodium-magnesium soil is nowhere near as stable as a sodium-calcium soil. I'm not sure if that answers your question.
Sort of, yeah. I think the biggest difference in what I've seen is that a lot of those tests have really high calcium percentages that are desirable in the exchangeables, and I suspect sometimes on our really acid soils that's not going to be possible unless you spend a ridiculous amount of money on lime.
Right. Yes. Well, the main thing about pH, I mean, that's why I said you've got to look at the process. If pH is regarded as your limiting factor, and the pH really only becomes seriously limiting when your pH is below 5.5 because that brings aluminium into solution. So if you're changing the pH from 6 to 6.5, you probably, I mean, that might give you a bit of a buffer against dropping the pH below 5.5, but I'm not sure that. So you've got to look at what your process, what are you trying to manage?
Yeah, well, that's it. I think because I quite often see tests where, to me, the pH is adequate for what people want to do, but then they'll say, oh no, but my exchangeable cations say I need more calcium. I'm going to go spend all this money on calcium. And I'm like, well, do you really need to?
Right.
And
Well, interestingly, even if you've got a sodic soil, the reason people add gypsum on a sodic soil is certainly to reduce the ESP, but the main effect of gypsum on a sodic soil is to increase the electrolyte concentration. And I tried to mention during the talk that if you have enough salt outside, then you won't get dispersion. And gypsum's a magical chemical because it actually dissolves just enough to provide stability to sodic soils, but doesn't dissolve enough to start causing salinity problems for plants. So that's why gypsum is so widely used, I guess, for stabilising sodic soils.
Yep, okay, thank you.
Thanks, Julian. And just out of interest, Julie, which part of New South Wales are you talking about?
North Coast. So one of our major labs up here tends to use the Albrecht's Rayment methods in their interpretations, and it can cause some good fun.
Okay, thanks very much, Julie. I have a message from Harry Kibler, and what he said is, I have organic matter of 10.8%. Is this a problem?
Yeah. Well, I guess my first question would be, is that organic matter, or I guess it has to be organic matter at 10.8%. To me, it's going to give you some advantages. It's probably a lot of that soil organic matter at 10.8%. A lot of that soil organic matter is probably going to be in what they call particulate soil organic matter. So it's probably not going to be adding that much. Like, you won't get 10.8% of the organic. All that organic matter is not going to be adding to your water holding capacity and aggregate stability and cation exchange capacity necessarily. A lot of it will be in the form of more or less decomposing organic material and particulate organic matter. However, what you can be sure of is that maybe something like 3 to 4% of that is actually contributing to your cation exchange capacity and your water holding capacity and various other things. So I wouldn't see it as a problem immediately. It depends also what texture it is. I presume it's reasonably loam or fine sandy loam to have that much organic matter. I don't know. Yeah, I wouldn't see it necessarily as a problem.
Okay, we've got a couple of comments here from Tony Cox. Tony says, hi, and a good presentation. The research coming out of the project that Tony's involved in, More Profit from Crop Nutrition, highlights low, medium and high production zones in paddocks, and we're looking at getting farmers to treat and sample these areas differently. Can you comment on that in terms of chemistry, please, Brian?
I think I know. I guess what Tony is asking is there soil chemistry causing the low production zones, the medium production zones and the high production zones? To be honest, it could easily be chemistry. It may also be just a change in soil type through the paddock, or often through paddocks you often get small wet zones as well, which are caused by just a small dip in the landscape, which makes them a lot wetter. And the wetter zones might have a high yield one year and maybe a lower yield the next, depending on the season, which makes it all very confusing to know exactly what's happening. So I would suggest that a lot of that would probably be caused by soil variability, which is related to probably clay content, parent material, just local landform, and different drainage conditions. It's hard to know without, but they will affect the soil chemistry, obviously. But probably it's a lot more the soil physical properties which are operating in some ways on a paddock, but they're through the effects of texture and waterlogging and drainage and so on. Yeah.
Great. So Brian, Tony's added another question in there where he said, how important do you think critical levels are in terms of crop development?
That's a good question, because some critical levels sometimes can be what they call a threshold, or they can be a continuous variation. The one which is the typical threshold is pH, because if your pH in water or drops below 5.5, or pH in calcium chloride drops below 4.7, you immediately know that you're going to start getting some aluminium 3+ in solution, which is going to have an immediate effect on your plant production, especially if you have a sensitive plant. On the other hand, if you're talking about perhaps nitrate levels or phosphate levels, or even bulk density levels, there's more of a continuous variation. So I guess the more nitrogen you have, often the higher the yields, until you reach a maximum, of course, until something else is limiting the plant. And the same with phosphorus. So some things are thresholds and some things are continuously variable. Yeah.
Great. Thanks, Brian. The next thing written I've got here is from John Laurie.
Yeah, I just wanted to make the point that magnesium can also be toxic, particularly on some soils high in iron, rocks high in, and magnesium can be a problem with soils with pHs above 5.5, especially canola. That was one thing that we discovered years ago, that you needed to have a higher pH to grow canola, and also lucerne needs higher pHs, so there is a need to put lime at pHs greater than 5.5 if you've got a magnesium toxicity problem.
Thanks, John.
Thanks, John. Yep.
So the next one's Michael Fitzgerald, and Michael has written a question in here because Brian, he says, how do you measure natural background soil organic carbon over time in a soil amended with an organic amendment?
I'll just interpret the question. I guess the question seems to be asking, how do you know what is your original native soil level of soil carbon compared to the soil carbon you've added since? To answer that question, when biomass and carbon is added to soil, essentially that carbon will be either metabolized and produced as CO2 by the microorganisms in the soil, or it will be assimilated into the soil organic carbon and the soil organic matter. So short of using something like an isotope, I'm not sure how you would try and distinguish between what was the native original soil carbon and the new soil carbon that you've added in. I think all the carbon atoms go together and get all mixed up. In fact, In fact, it's probably wrong to say that soils bank carbon, because the carbon goes in, but that carbon has been continually reprocessed and processed by the microorganisms and plants and so on. So it's not like the carbon just sits there. It's actually being churned around and moved from microorganisms to soil organic matter, back to microorganisms, and then back to soil organic matter. So it all gets churned up and it all becomes one as soil organic matter. So I'd say the only way to do that could be with isotopes, but the only other way would be if you had a paddock next door that you knew was a native, undisturbed site. Maybe you could just monitor what the soil carbon level was on that paddock next door and say, okay, this is what the soil carbon level would be if we hadn't added all that soil organic matter.
Great. All right, so Glenn Yerbegang has got a written question here.
I'm just wondering whether you have a comment on micronutrients, like which ones are the most significant with soil tests and which can be accurately determined by soil tests rather than leaf tissue tests.
I guess you're interested in how the micronutrients would affect the plant growth, but certainly molybdenum is certainly an important one for legumes and so on.
Do you think we can trust the results that we get from a soil test? Are they accurate?
I guess it comes back to what the experience has been with, and I guess my knowledge of some of testing for micronutrients isn't that strong. What I would probably recommend to you is that you go and read the book by Peveril, where he lists molybdenum, all the different tests and field studies for molybdenum, also for boron. He also lists ones for cobalt. So that probably doesn't answer your question that well, but certainly the book by Peveril has all that sort of information in it.
No worries. Thank you.
Yep.
Thanks, Glenn. Harry Kibler. So Harry's question is, I grow native flowers that are susceptible to P, phosphorus. What is the best way to supply low amounts of phosphorus over a long period? Do you want the question again, Brian?
Yeah, no, I think the obvious way is with organic materials, because and the organic material, you probably need to get some organic materials where you know what the carbon to phosphorus ratio is, and also the carbon to nitrogen ratio, because the carbon nitrogen ratio is important, because the carbon nitrogen ratio will determine how rapidly that organic matter decomposes. So if you want to make it slow release, you probably want to have some organic materials which are perhaps have some phosphorus, but the carbon nitrogen ratio is not too high. I mean, there could be some slow release type phosphorus fertilizers available on the market as well, but certainly organic materials which don't decompose too rapidly, and that's why you'd leave out chook manure and things like that, because they have so much nitrogen and that they'll decompose rapidly. But
Okay.
but the right compost would probably do the job, unless they've got some specialised fertilisers.
Terrific. Phil Hurst doesn't have a microphone.
Right. Oh, good. Yeah.
Yep. And Phil has written, hi, Brian, a question on CEC or cation exchange capacity in acid sulfate soil. By adding high aluminium values, the results end up quite high. Is this a good indication, is his question?
I think determining soil properties on acid sulfate soils is a specialised field and requires specialised tests. So measuring cation exchange capacity on acid sulfate soils probably depends an awful lot on whether it's been drained and what the pH is and various other things. So I guess if he sends me an email, I can probably pass him on to some people who know much more about that kind of details on that sort of. Acid sulfate soils are, as you realise, because they have the sulphur, they have iron sulfides, and when you drain them, they start producing sulfuric acid. So exactly how you determine things like cation exchange capacity and so on, I'm I think you'd want somebody who knows, actually specialises in acid sulfate soils to make a comment on that. Yes.
Okay. And
But
Phil,
if you send me an email, I can put you on to the person or pass you on to the manual as well, which talks about soil tests for acid sulfate soils. Yeah.
That'd be good. And Phil, as you probably know, has a long background in acid sulfate soil, so yeah, it's getting pretty technical. But thanks for the question, Phil. Okay, Tony Cox has made another comment. Is ensuring your lab has ASPAC certification is a good start to ensuring your tests are accurate. And Harry Kibler has said, thanks for your answer. And with high organic matter, would increasing the soil microorganisms add to the CEC?
I suspect adding soil if your soil organic matter is already high, adding soil microorganisms probably will not add to the CEC, because the CEC requires the negative charge from the soil organic matter, and it's a negative charge from the soil organic matter which is important in providing CEC, and therefore soil microorganisms themselves are probably not going to do that. Plus, soil microorganisms and the percentage of the soil organic matter that living organisms make up is normally fairly small, possibly around about 3 to 5% of the overall soil organic matter, or maybe a bit less. Yeah. So I wouldn't. Generally, I would think adding organisms is probably not going to directly increase the cation exchange capacity. Yeah.
Yep. Great. Thank you so much, Brian, for a really informative webinar, and great to have you along. It's again broken the record for attendees. So for another fantastic webinar, thanks again to you, Brian. So thank you everybody for attending.
Okay, right. Thank you everyone. Yeah.
Fantastic. I'll bring it to a close now.